All right, so in this one, we're going to talk about cell transport. Our essential question, or what I want you to know after this talk, is how food, waste, and water move in and out of the cell. So you imagine the cell needs to not only produce energy and produce macromolecules, but they need to be able to take in food from outside, like because we eat, right, in order to create macromolecules like proteins. Um, it also produces waste, and so it has to get rid of that waste somehow, and then it also has to take in water. And so, how does this occur? Is the question that we kind of have to ask ourselves, and hopefully we'll answer that today. So first, if we think about it, the cell membrane is kind of the gatekeeper, right? It's on the edge of the. It's basically what holds the cell together. And so within the cell membrane is the cytoplasm, right? So if I just drew an animal cell, right? So within here is the cytoplasm. So it holds the cytoplasm, and it also acts as a barrier to the outside of the cell. But there's got to be things that can come in and out. And so it's kind of like a gatekeeper. So it's not like a wall where there's no movement inside and outside. Which is what we call semi-permeable. So some material can transfer, but not all. So it's selective. So we can say semi-permeable, we can say selectively permeable. And the cell membrane is mostly made of lipids. Remember, we have this is the glycerol. And then this is the fatty acid chain. And so we call them phospholipids. Phospho refers to, these are kind of some special lipids that have an extra component called a phosphate group. And so we call them phospholipids. And then there's also proteins that help with the movement of things in and out. So cell membrane, it's the gatekeeper. Just remember it's semi-permeable, and it's made of lipids and proteins. So to move things in and out, there's two types. One's called passive, and the other one's active. So passive, if you think of somebody who's passive, they kind of just sit there. They're not really using energy. So passive transport is movement of material across the cell membrane without using energy. There's two types of examples, diffusion and osmosis. And then active transport is when you need, like, if you think of something active, you need energy. So there are protein pumps. Proteins that sit in the cell membrane, they kind of move things back and forth. So one type of passive transport is called diffusion, and that's when you move movement of molecules from an area of high concentration to low concentration. So if I dump say sugar in this case, what happen is that the sugar will diffuse out. So they're spread out equally until it reaches equilibrium. Equilibrium is kind of when they are all kind of spread out equally apart from each other. You can think of this as well, like if somebody sprays perfume, what happens is people closest will start smelling it, but then eventually, as the particles diffuse out, what happens is more and more people smell it. Why? Because the particles are diffusing away. Another type of passive transport is called osmosis. And that's instead of the particles diffusing out, in this case, we will have the water itself. And so it's actually a good time to introduce a concept. So there's something called a solute, and then solvent. So like a solute is what is being dissolved. So a common example of a solute is like salt or sugar or something. And then the solvent is what is dissolving. So a common example is water. So if I put salt in water, if I dissolve salt in water, salt is a solute, and then water is a solvent. All right, so what is osmosis? Instead of the solute or the salt moving, the water is the one that can only pass through the cell membrane. It's actually it's the one that will move. So if we look here, it says, what direction do you think the water will flow? Well, here there is a higher concentration, and then here there's a lower concentration. Remember, the goal of any type of passive transport is you're going to go from the water will want to kind of reach equilibrium. They want to be equal. But instead, what won't move are these individual solute malt. So this can't move. Only the water can move. And so the water will actually move in this direction. It'll move in that direction because it will want to balance out the amount of the concentration. Let's just move to the next slide, and then maybe you'll understand further. So here's just tricks to remember movement of water in osmosis. So salt sucks, or you can say the solute sucks. What that means is the area with higher concentration or amount of solute will always suck the water from that area. Or you could say water follows salt. So water will always go to the area with higher concentration of solute. So let's just use the example here. So which area has a higher? So I'm here. Which area has a higher concentration or amount of salt? Well, it would be this area, right? So the water follows the salt, or the salt sucks. So which way would the water flow? Well, the water would flow where there's more salt. Okay? And so what would happen is it would try to reach equilibrium. Oh, let me just go back real quick. Oops, wrong way. So what would happen here is you can see the water moved in this direction, right? And so now you can kind of see they're at equal concentration. What that means is there's the same amount of salt per water content here, right? And so that's what we call equilibrium. Okay. So remember, water follows salt, or salt sucks. Okay. So there's three types of tonicity of solution. So tonicity refers to the concentration. So concentration is the amount of solute per solvent. So it's the amount of salt per water in our example. So if you're isotonic means that it's 10% salt, 10% salt. So it could be 50% salt, 50% salt, as long as they're the same. Iso means the same. Which way will the water flow? Well, are they balanced? Are they in equilibrium? Yes. So if one flows this way, you'll have flow in both directions. What about if the solution is hypotonic? So hypo means less. So in this case, the solution so this is supposed to be a cell, or anything with a semi-permeable membrane. So imagine if this was a cell, so this is a cell. So we have 20% salt. Well, which way would the water flow? Well, remember, water flows, so either salt sucks or water follows salt. So water would go to where there's more salt. Well, where there isn't more salt? Well, in the cell, there's 20% salt as opposed to 10. So water would flow into the cell. What do you think would happen to the cell? Well, if water went into the cell, the cell would expand, even getting to the point that it could perhaps explode, right? Now, a hypertonic is the opposite. Hyper means more, right? Like kids hyper, they're really moving around like crazy. So that means there's more solute or a higher concentration on the in the solution. So in here we have 20% and only 10% in the cell. Well, remember, water follows salt, so water's going to go out. What would happen to the cell? The cell would shrivel. So if you guys remember, when we looked at the leaf cell and we added a bunch of salt, this is what happened, right? There was a lot of salt outside the cell. So what happened was the water escaped. The water left the cell and went onto the slide from the leaf cell, the Lodia cell, and so we saw that the vacuoles inside the cell shrunk, and as well as the vacuoles themselves shrunk. The cells shrunk themselves, right? And so this is what happens when you put cells in a hypertonic solution. So I have a couple of questions. You could try these on your own. It says, what would happen to a cell that was in a hypertonic solution? Draw what would happen. So let's just think back. A hypertonic is where hyper means more, right? And so if this was 10%, then hypertonic would be like 20%, right? So which way would the water flow? It would flow out. So what would happen to the cell? The cell would shrink. It says a cell bursts or exploded due to the tonicity of a solution. Was the solution hypertonic, hypotonic, or isotonic? Well, if a cell bursts, which way was the water flowing? It was flowing this way, right? Well, the only way that it flows into a cell is if the cell had higher, like say 30% salt, and out here was, I don't know, just make it 15%, right? So in this case, was the solution hypertonic, hypotonic, or isotonic? Well, when the solution is less, that's hypo. Hypo means less. Okay? The last one is active transport. So active transport, so the last two we've been doing is diffusion, osmosis. That's an example of passive transport. Like if you have a semi-permeable membrane, like in the cell, this will automatically happen. It doesn't require energy. But some of them, when let's say you want to move something against the concentration gradient, so you want to move something against passive transport, or what would naturally happen in osmosis or diffusion, you have to actually provide energy. And so there are proteins within the cell membrane that allow energy. The really easy example is, when everybody's exiting a building at the beginning of a passing period, right? Everybody's leaving their classes, so the flow is one way, right? And so if you are early to class and you're trying to get to class, you are going against the flow, right? And so that requires a lot of energy. And so that's kind of like active transport when you're moving against the flow. So I would remember that active transport requires energy, and it's against the concentration gradient, and there are special proteins and pumps found in the cell membrane to do that.